Welcome, everyone, to tonight's exploration into the mesmerizing world of computational chemistry, specifically focusing on molecular dynamics. As we settle in for a relaxing journey through the microscopic realm, we'll uncover the secrets of how molecules move and vibrate within our bodies, all in slow motion. Imagine shrinking down, far beyond the limits of our everyday perception, to witness the intricate dance of atoms and molecules that underpin life itself. Tonight, we'll not only visualize this dance, but also understand the computational techniques that allow scientists to simulate and study these processes. Our focus tonight will be on molecular dynamics, MD, a powerful simulation technique used to study the physical movements of atoms and molecules. At its core, MD is based on solving Newton's equations of motion for a system of interacting particles. These particles, representing atoms or groups of atoms, are subjected to forces derived from interatomic potentials or force fields. By iteratively calculating the forces acting on each particle and updating their positions and velocities over time, we can simulate the dynamic behavior of the entire molecular system. This allows us to observe and analyze how molecules fold, interact, and react under various conditions. To truly appreciate the significance of molecular dynamics, let's take a moment to consider the sheer complexity of the molecular world within our bodies. Our cells are filled with countless proteins, enzymes, nucleic acids, and other molecules, each constantly vibrating, rotating, and colliding with one another. These movements are not random, but are instead governed by the laws of physics and chemistry. Understanding these dynamics is crucial for comprehending how biological processes such as protein folding, enzyme catalysis, and drug binding occur. Imagine a single protein molecule, a complex chain of amino acids, folding into its unique three-dimensional structure. This folding process is not instantaneous, but rather a dynamic journey guided by various forces, including electrostatic interactions, hydrogen bonds, and hydrophobic effects. Molecular dynamics simulations allow us to visualize this folding process in atomic detail, revealing the intermediate states and pathways that the protein molecule traverses as it reaches its final functional form. This knowledge is invaluable for understanding protein misfolding diseases such as Alzheimer's and Parkinson's, where proteins fail to fold correctly and aggregate into harmful structures. Now, let's delve into the mathematical and computational foundations of molecular dynamics. The fundamental equation underlying MD simulations is Newton's second law of motion, which states that the force acting on an object is equal to its mass times its acceleration. F equals ma. In the context of molecular dynamics, we apply this law to each atom in the system. The force acting on an atom is determined by the potential energy function, which describes the interactions between atoms. This potential energy function is often referred to as a force field, and it includes terms that account for bond stretching, angle bending, torsional rotations, and non-bonded interactions such as van der Waals forces and electrostatic interactions. The choice of force field is crucial for the accuracy and reliability of molecular dynamic simulations. There are several commonly used force fields, each with its own strengths and limitations. Some popular force fields include amber, CHARMM, and Gromos. These force fields differ in their parameterization and functional forms, and the selection of the appropriate force field depends on the specific system being studied and the desired level of accuracy. For example, amber is often used for simulating proteins and nucleic acids, while CHARMM is commonly used for simulating lipids and carbohydrates. Once we have chosen a force field, we can begin the process of setting up and running a molecular dynamic simulation. The first step is to define the initial coordinates and velocities of all the atoms in the system. This information is typically obtained from experimental data such as X-ray crystallography or nuclear magnetic resonance, NMR spectroscopy. Alternatively, we can use computational methods to generate an initial structure, such as homology modeling or de novo structure prediction. Next, we need to define the simulation box, which is a rectangular or cubic volume that contains the molecular system. The size of the simulation box should be large enough to accommodate the system without introducing artificial constraints. Periodic boundary conditions are often applied to the simulation box to mimic an infinite system. This means that when a molecule leaves the box on one side, it re-enters on the opposite side. Periodic boundary conditions eliminate surface effects and allow us to simulate bulk properties. With the initial coordinates, velocities, and simulation box defined, we can now run the molecular dynamic simulation. The simulation proceeds in a series of discrete time steps, typically on the order of femtoseconds, 10 to the power of -15 seconds. At each time step, we calculate the forces acting on each atom and update their positions and velocities using a numerical integration algorithm. The most commonly used integration algorithm is the Verlet algorithm, which is simple, efficient, and time reversible. As the simulation progresses, we can monitor various properties of the system, such as temperature, pressure, and energy. These properties should remain relatively constant throughout the simulation, indicating that the system is in equilibrium. If the system is not in equilibrium, we may need to adjust the simulation parameters or run the simulation for a longer time. One of the key challenges in molecular dynamic simulations is the computational cost. Simulating the dynamics of large molecular systems over long time scales requires significant computational resources. The number of calculations increases dramatically with the number of atoms in the system and the length of the simulation. To overcome this challenge, researchers have developed various techniques to accelerate molecular dynamic simulations. These techniques include parallel computing, GPU acceleration, and coarse-grained modeling. Parallel computing involves dividing the simulation among multiple processors or computers, allowing the calculations to be performed simultaneously. GPU acceleration utilizes the massive parallelism of graphics processing units, GPUs, to accelerate the force calculations. Coarse-grained modeling reduces the number of degrees of freedom in the system by representing groups of atoms as single beads. This reduces the computational cost but also sacrifices some level of detail. Now, let's consider some specific examples of how molecular dynamic simulations are used to study biological systems. One important application is in drug discovery. Molecular dynamic simulations can be used to screen potential drug candidates by simulating their interactions with target proteins. This allows researchers to identify compounds that bind strongly to the target protein and inhibit its function. Molecular dynamic simulations can also be used to optimize the structure of drug candidates to improve their binding affinity and selectivity. Another important application is in understanding the mechanisms of enzyme catalysis. Enzymes are biological catalysts that accelerate chemical reactions in cells. Molecular dynamics simulations can be used to study the dynamic motions of enzymes and substrates during the catalytic process. This can provide insights into the factors that determine the rate and specificity of enzyme reactions. Molecular dynamic simulations are also used to study the folding and aggregation of proteins. Protein folding is the process by which a protein molecule acquires its unique three-dimensional structure. Protein aggregation is the process by which protein molecules clump together to form large aggregates. Both protein folding and aggregation are important in various biological processes and diseases. Molecular dynamic simulations can be used to study the factors that influence protein folding and aggregation, such as temperature, pH, and ionic strength. Let's take a moment to visualize a molecular dynamic simulation of a protein folding. Imagine a long chain of amino acids, initially unfolded and disordered. As the simulation progresses, the chain begins to fold, guided by various forces. Hydrogen bonds form between different parts of the chain, pulling them closer together. Hydrophobic amino acids cluster together in the interior of the protein, away from the surrounding water molecules. Gradually, the protein molecule acquires its final three-dimensional structure, with its active site perfectly positioned to bind to its substrate. As we continue our journey through the world of molecular dynamics, let's explore some of the advanced techniques that are used to enhance the accuracy and efficiency of simulations. One such technique is enhanced sampling, which aims to overcome the limitations of traditional molecular dynamic simulations in exploring rare events and conformational transitions. Enhanced sampling methods include umbrella sampling, metadynamics, and replica exchange molecular dynamics. Umbrella sampling involves applying a biasing potential to the system to encourage it to visit specific regions of conformational space. This allows us to sample rare events that would not be observed in a standard molecular dynamics simulation. Metadynamics involves adding a history-dependent potential to the system that discourages it from revisiting previously sampled states. This allows us to accelerate the exploration of conformational space and identify new metastable states. Replica exchange molecular dynamics involves running multiple simulations at different temperatures and periodically exchanging the configurations between them. This allows us to overcome energy barriers and sample a wider range of conformations. Another important technique in molecular dynamic simulations is free energy calculation. Free energy is a thermodynamic quantity that determines the equilibrium distribution of states in a system. Calculating free energies is essential for understanding the stability of different conformations and the driving forces behind molecular processes. There are several methods for calculating free energies, including thermodynamic integration, free energy perturbation, and Bennett acceptance ratio. Thermodynamic integration involves integrating the derivative of the free energy with respect to a parameter along a reversible path. Free energy perturbation involves calculating the free energy difference between two states by perturbing the system from one state to the other. The Bennett acceptance ratio is a statistical method that combines data from multiple simulations to estimate the free energy difference between two states. Now, let's delve into the challenges and limitations of molecular dynamics simulations. One major challenge is the accuracy of the force fields. Force fields are empirical models that approximate the interactions between atoms. They are parameterized based on experimental data and quantum mechanical calculations. However, force fields are not perfect and can sometimes lead to inaccurate results. The development of more accurate and reliable force fields is an ongoing area of research. Another challenge is the timescale limitations. Molecular dynamics simulations are limited by the time step and the length of the simulation. Simulating long time scales requires significant computational resources. This limits our ability to study slow processes such as protein folding and aggregation. Enhanced sampling techniques can help to overcome this limitation, but they are not always applicable. Despite these challenges, molecular dynamics simulations have become an indispensable tool for studying biological systems. They provide insights into the dynamic behavior of molecules that cannot be obtained from experiments alone. As computational power continues to increase and new simulation techniques are developed, molecular dynamics simulations will play an even greater role in advancing our understanding of the molecular world.